There are many problems in wound care that requires our attention as future healthcare workers. One of them is the rise of antibiotic resistance in wound infections. One of the major problems in wound care today is the increase in antibiotic-resistant bacteria such as Staphylococcus aureus, Pseudomonas aeruginosa, and Escherichia coli. Conventional antibiotics are becoming less effective, leading to delayed healing, prolonged hospital stays, and higher risk of complication. The second problem is limitation of conventional wound dressings. Traditional wound dressings mainly act as physical barriers, but some contain antimicrobial agents. They may cause cytotoxic effects to healthy cells, lose effectiveness over time, and failing to penetrate bacterial biofilm, which protect bacteria from antibiotics and immune response. The third problem is biofilm information in chronic wounds. Bacteria in chronic wounds often form biofilms, which are structured communities of bacteria embedded in a protective matrix. Biofilm reduce antibiotic penetration, increase bacterial resistance, and maintain persistent infection and inflammation.
To address this issue, we propose Phage-Aid. Phage-Aid is a hydrogel wound dressing infused with bacteriophages, which are viruses that specifically infect and destroy bacteria. The hydrogel maintains a moist wound environment, while the bacteriophages actively target harmful bacteria at the infection site. Unlike antibiotics, bacteriophages are highly specific. They attack only the pathogenic bacteria without harming the human cells or beneficial microbiota.
For the scientific basis of this product, Phage-Aid combines two key components, which is bacteriophage and hydrogel wound dressing. Both components play a specific role in combating infection and promoting wound healing. The first component, bacteriophage, are viruses that specifically infect bacteria. Their mechanism of action is highly targeted and involves the following steps. The first step is adsorption, where the bacteriophage binds to specific receptors on the surface of the target bacteria. The second step is injection of genetic material, where the phage injects its DNA or RNA into the bacterial cell. The third step is application, where the phage hijacks the bacterial machinery to produce new phage particles. The last step is bacteriolysis, where the bacterial cell bursts, which will release new deformed phage that go on to infect nearby bacteria. The second component, hydrogel dressing, serves as a delivery platform and wound healing support system, where it maintains a moist wound environment which promotes cell migration and tissue regeneration. It also protects the wound from external contamination and allows control and sustained release of bacteriophage, which also provides mechanical stability while being non-adhesive and biocompatible. Hydrogels can be made from materials such as alginate, chitosan, gelatin, or polyethylene glycol.
For our marketing strategy, Phage-Aid is marketing directly to the hospital through the trained medical representative and also representative educate clinician on science, safety, and cost effectiveness of phage therapy, and also demonstration and clinical trials data are used to support both purchasing contract of hospital and care centers and long-term partnership with healthcare providers ensure consistent supply and feedback driven important. For our business feasibility, we are gonna tell you about how Phage-Aid is manufactured and marketed. Firstly, on our laboratory production of macrophage, bacteriophage are isolated from natural sources and selected for their ability to target the specific pathogen of the bacteria. Also, each phage undergoes strict screening to ensure safety, specificity, and effectiveness. Also, phages are cultured using their bacterial host under controlled laboratory conditions, and after replication, phages are purified to remove bacterial debris and also endotoxin. Quality control also ensures consistent potency and stability of the phage preparation. We undergo the integration into the hydrogel dressing, purified bacteriophage incorporated into a biocompatible hydrogel matrix. Hydrogel protect phages from the environmental threats while allowing gradual release at the wound site. The dressing is sterilized using method that preserve phage viability. Also, the final product are sealed and sterile in packaging and labeled for clinical use. Manufacturing follows good manufacturing practice standards to ensure safety and reproducibility. Last but not least, how Phage-Aid saves life endings is by addresses a clinical gap in a modern medicine where antibiotics are no longer effective. Also, by selectively destroying the antibiotic-resistant bacteria, it prevents wound infection from processing to sepsis. Also, early and effective infection control significantly reduces the need for the limb amputation, especially in diabetic patients. The localized nature of treatment minimizes the systemic side effect and preserves beneficial bacteria. Faster wound healing, shorten the hospital stay, and improvement patient outcomes. Overall, Phage-Aid contributes to better survival rate, improved quality of life, and reduce healthcare costs. And also, this product represents a sustainable and innovative approach.
In conclusion, Phage-Aid represents an innovative and timely solution to one of the most pressing challenges in modern healthcare, which is the increasing threat of antibiotic-resistant wound infections. By combining the targeted antibacterial action of bacteriophages with the protective and healing properties of hydrogel dressings, Phage-Aid goes beyond conventional wound care approaches. Rather than relying solely on antibiotics, this product works directly at the infection site, targeting harmful bacteria while preserving healthy human cells and beneficial microorganisms. From a medical microbiology perspective, Phage-Aid demonstrates how fundamental scientific concepts can be translated into practical healthcare solutions. It applies bacteriophage biology in a way that is both clinically relevant and aligned with current efforts to reduce antibiotic overuse. In terms of impact, Phage-Aid has the potential to improve wound healing outcomes, reduce treatment duration, lower healthcare costs, and support global strategies to combat antibiotic resistance. It also opens opportunities for further development, such as customizing bacteriophage combinations for different types of bacterial infections. Overall, Phage-Aid highlights the importance of innovation at the intersection of science, healthcare, and technology. With further research and development, this concept could contribute meaningfully to safer, more effective, and more sustainable wound management practice in the future. Thank you for your attention.